A 5-month-old infant is admitted to the emergency department. The triage notes describe poor sucking ability, a sudden loss of head control, and rapidly fading motor skills. Blood work reveals severe lactic acidosis. Genetic testing points to Lee disease, an extremely rare mitochondrial disorder. The infant's pyruvate dehydrogenase enzyme complex is failing. Without this functional enzyme complex, the cell cannot achieve oxidative phosphorylation. Unable to oxidize pyruvate into acetyl-CoA, the body is forced to ferment it into lactic acid, starving the brain of energy. Biological life relies on a continuous, highly controlled transfer of electrons. When a chemical species loses electrons, it undergoes oxidation. When it gains electrons, it undergoes reduction. The law of conservation of charge dictates that an isolated loss of charge is physically impossible. If one atom is oxidized, another must be simultaneously reduced. Identifying where a metabolic pathway has collapsed or quantifying a reaction yield in the lab depends on tracking the exact sequence of electron movement from one species to the next. To map electron ownership across complex molecular bonds, chemistry uses a standardized mathematical system. This carbon dioxide diagram models formal charge, assuming perfect one-to-one electron sharing. Oxidation state assumes unequal sharing, awarding bond electrons to the most electronegative element. Oxygen takes the electrons, taking an oxidation state of -2, leaving the central carbon at +4. To assign these states efficiently, we follow a strict hierarchy of rules. First, any free, unbonded element always carries an oxidation state of 0. When bound in compounds, group 1A elements are assigned +1, and group 2A elements take +2. We then anchor the remaining elements using highly electronegative atoms. Halogens generally carry a -1, and oxygen almost always takes -2. Finally, the sum of all individual oxidation states must exactly equal the net charge of the overall molecule or polyatomic ion. Applying these rules to tin chloride and lead chloride, we isolate the halogens. Tin's oxidation state increases from +2 to +4, acting as the reducing agent. Lead state decreases from +4 to +2, serving as the oxidizing agent. By systematically mapping these states, we identify the flow and track the exact electron transfer driving any stable chemical interaction. In volatile aqueous environments, tracking electrons by inspection alone breaks down. Transient species like water and free protons constantly alter the system's mass and charge. We cannot balance this permanganate and iodide reaction by inspection. Step 1 divides the equation into oxidation and reduction streams. Step 2 balances atoms, doubling the iodide reactant, adding 4 water molecules to the manganese product side, and balancing hydrogen with 8 protons. Step 3 neutralizes charge by adding 2 electrons for iodine and 5 for manganese. We find the lowest common multiple, scaling iodine by 5 and manganese by 2. The merged master equation cancels out the 10 overlapping electrons, leaving a perfectly balanced net ionic equation. The half-reaction method is a mathematical necessity. It forces the system to balance electrical charge first, ensuring absolute conservation before recombining the chemical mass. There is a specific structural edge case that disrupts standard redox assumptions: disproportionation. In a disproportionation reaction, a single element acts as both the oxidizing and reducing agent. It is simultaneously oxidized and reduced, splitting its oxidation states across multiple products. Biology utilizes this chemical anomaly as a vital defense mechanism. Cells deploy an enzyme called superoxide dismutase to neutralize toxic free radicals. This diagram shows a reactive oxygen-free radical entering the enzyme's active site. Because the negative charge is shared across 2 oxygen atoms, the element carries an unstable fractional oxidation state of -1/2. Inside the active site, copper and zinc atoms act as reducing agents to force a dismutation. The radical disproportionates. One path reduces the oxygen to -1, forming hydrogen peroxide. The other path oxidizes it to 0, producing stable molecular oxygen. Biology engineers disproportionation as a targeted failsafe. By leveraging the extremes of electron transfer, enzymes disarm cellular threats before they cause systemic tissue damage. We move from biological theory to the practical reality of precise laboratory quantification using oxidation-reduction titrations. While acid-base titrations track the movement of protons, Potentiometry utilizes a voltmeter to track the exact transfer of charge through changes in the solution's voltage. A specific, highly visual application is an iodometric titration. Here, a dark starch indicator solution snaps to completely colorless to signal that a sodium thiosulfate solution has reached its equivalence point. This instantaneous shift from blue-black to colorless suggests a simple exchange, but the stoichiometry is governed by a multi-step sequence. This equation reveals the first true underlying step. Iodate reacts with iodide to generate triiodide anions. In the second sequential step, those newly formed triiodide ions are reduced by the thiosulfate. The algebraic link between these two reactions is the intermediate triiodide. To algebraically consume all 3 triiodide ions generated in the first step, we must multiply the entire second equation by 3. Only after scaling the intermediate triiodide does the actual stoichiometric ratio emerge. 1 mole of iodate requires exactly 6 moles of thiosulfate. Failing to decouple and appropriately scale these half reactions results in a mathematically flawed calculation, rendering the laboratory data useless. The exact same electron flow that dictates the precise standardization of analytical reagents also governs the onset of neonatal metabolic failure. The required protocol remains consistent. Identify the baseline oxidation states, strictly isolate the half reactions, and balance the electrical charge before reconciling the mass. Mastering oxidation reduction requires wielding the universal language of electron transfer, this silent currency that dictates both chemical analysis and biological survival.